Manuela Avendaño, and today I will be presenting this article that is about dissecting the role of tyrotropin in the DNA damage response in human thyrocytes after iodide and gamma radiation and hydrogen peroxide. Okay, so tyrotropin. This hypothalamic hormone is in charge of activating the thyroid gland for the production of T24. And the elevated serum levels of this hormone can cause papillary thyroid cancer or PTC. DNA damage is a problem in the organization of the DNA that can be caused by metabolism radiation as beta and gamma radiation or toxic substances as ROS. For example, we have hydrogen peroxide that can induce double strand DNA breaks and chromosomal rearrangements. We also have gamma radiation that can increase the production of ROS mediated by duoxide protein. And we have iodide that is a thyroid mutagenic agent that appears in nuclear accidents. This three can cause papillary thyroid cancer. Okay, so this is basically what they did in the study. They used TSH to activate these two proteins, these G proteins. So this GQ protein activated the inositol triphosphate to produce ROS as hydrogen peroxide. This causes DNA damage. And we also have TSH that increases the function of the GS protein that activates cyclic AMP as forsoclin, and they induce proliferation of the thyrocytes. And this, that is, bovine cellulose can also activate the proliferation. The P21 protein, it's a protein that can measure the levels of cellular division and can inhibit the proliferation. And we also have that cyclic AMP increases iodide uptake, and this with beta and gamma radiation can cause DNA damage. We have to remember that NAC or NAC is an antioxidant that can inhibit the effect of TSH in GQ. The objective is to analyze the role of tyrotropin in the DNA damage response in human thyrocytes after iodide, gamma radiation, and hydrogen peroxide. Thyrocytes were obtained from patients undergoing total or partial thyroidectomy, and the basis is to prepare human thyrocytes, and they make a digestion and seeding in a nutritive medium. So the utility is to allow the manipulation of the cells and make it easier to submit this culture to DNA damaging agents. Western blot is an electrophoresis technique that is used for the detection of specific proteins according to their molecular weight, and they use antibodies. It was used to detect the P21 protein and also the caspase-3 protein levels in the thyrocytes after they were exposed to the agents. The sequenciation consists of determining the RNA sequence based on a DNA sequence, and it was used to observe the gene expression after they induced the DNA damage. Okay, so the immunofluorescence is basically a staining, so they can observe how much of the DNA has been damaged according to the coloration. And it was made to compare the surface of damaged DNA with the rest of the surface. Okay, so here in this result of the staining, we can observe that tyrotropin had an increased DNA damage in non-exposed and also in the exposed cells, but it didn't have an effect on the gamma radiation or the H2O2 induced DNA damage. Other agents as forsoclin and EGF did not have an effect on the induced DNA damage. We can conclude this because of the color of the pink coloration we see. Figure 5 shows the gene expression. This really is read by a software, but we can see that it is divided in 2 different hour lapses. It shows that the influence of TSH was most evident in the 24-hour lapse, Also showed that the 4-hour response included alteration in the expression of genes involved in apoptosis, inflammatory response, and cell adhesion. And the 24-hour response included alteration in the expression of genes involved in DNA replication, evolution of the mitotic cycle, cell proliferation, apoptosis, and DNA repair. On figure 6, we can observe a significant decrease in the proliferation rate after the exposure to iodide and to the gamma radiation, always being compared to the control. And the difference was only significant in the TSH-treated cells. We can also observe that H2O2 didn't have a significant influence in the thyrocyte proliferation. Western blotting. First of all, let's remember that P21 protein causes the cell cycle arrest, and the caspase-3 is in charge of cellular apoptosis. On figure A, we can observe that P21 protein has a weight of 20 kilodaltons, and that it appears when the sample is exposed to gamma radiation, and it appears even if it is exposed to TSH or not exposed to TSH. So it has a significant increase in the intracellular amounts of the protein, non-depending of the presence of the TSH. On figure B, we can observe that caspase-3 had a molecular weight of 18 kilodaltons, and that they did two samples, one of primary human thyrocytes and another one that they use as a positive control. We can see that the protein only appeared on the control, which means that the caspase-3 wasn't detecting in the primary human thyrocytes that were submitted to the radiation and to pseudosphain. This means that in this sample there was no sign of apoptosis. So first author was making a study on the sodium iodide importer on mice with hypothyroidism. He found out that the increased levels of TSH that we can find in people and also mice with hypothyroidism can increase the levels of ROS, which obviously increases the levels of H2O2. This author also found out that this happens with the parallel downregulation of these antioxidant genes. This information matches with what they say in the article. The second author says that iodide can provoke DNA DSB, which means DNA double-strand breaks formation in the human thyrocytes, which means DNA damage. Iodide causes DNA damage, and this is proved in the article. The third author said a significant delay in gamma radiation-induced DNA damage repair was observed in human thyrocytes previously exposed to H2O2. This information doesn't match with what they say in the article, because in the article they say that TSH doesn't have an influence on the damage repair,